Okay, class, today we are covering the history of DNA, so we can move on to more in-depth discussion about DNA regarding transcription and translation. The molecule that is the basis for heredity is called deoxyribonucleic acid, or DNA, and it contains the patterns for constructing proteins in the body. A new understanding of heredity was possible once it was determined that DNA consists of two chains twisted around each other, or double helices, of alternating phosphate and sugar groups, and that the two chains are held together by hydrogen bonds between pairs of organic bases, adenine A with thymine T, and guanine G with cytosine C.
Wait, so who's actually responsible for discovering DNA? I thought that scientists have always known about this part.
It is a common misconception that James Watson and Francis Crick discovered DNA in the 1950s. In reality, DNA was discovered decades before, yet it had not been widely accepted. It was by following the work of the pioneers before them that Watson and Crick were able to come to their groundbreaking conclusion about the structure of DNA in 1953. To tell this story correctly, we also need to introduce Rosalind Franklin and Maurice Wilkins, who are also very essential to this story. So, what we do know is that Chargaffs concluded that almost all DNA, no matter what organism or tissue type it comes from, maintains certain properties, even as its composition varies. In particular, the amount of adenine A is usually similar to the amount of thymine T, and the amount of guanine G usually approximates the amount of cytosine C. In other words, the total amount of purines A plus G and the total amount of pyrimidines C plus T are usually nearly equal.
Yes, indeed. And that absolutely has something to do with the structure.
Hi, Maurice!
Hi guys, how's your research going?
It's going great, for I feel like we are on the brink of a breakthrough.
I think I may have something that can help. There's this colleague in my lab that you might know, and her name is Rosalind Franklin. Although I would never say this to her directly, she is a very good with X-ray photographic work. She was able to capture this image that looks important, and I took it without her knowing so we could use it. And I figured she wouldn't mind.
This evidence has demonstrated that the two sugar phosphate backbones lay on the outside of the molecule. Crick, this is phenomenal!
Wait, so what happened after that? Who received credit for this discovery?
Watson and Crick's solution was formally announced on April 25, 1953, following its publication in that month's issue of Nature magazine. Their article revolutionized the study of biology and medicine. When Crick and Watson won the Nobel Prize in 1962, they shared it with Wilkins. Franklin, who died in 1958 of ovarian cancer, was thus ineligible for the award because they cannot give the award to someone that is dead. She never learned of the role her photos played in the historic scientific breakthrough. Watson and Crick were not the discoverers of DNA, but rather the first scientists to formulate an accurate description of the molecule's complex double helix structure. Watson and Crick's work was directly dependent on the research of numerous scientists before them, including and not limited to Friedrich Miescher and Erwin Chargaff. Thanks to researchers such as these, we now know a great deal about genetic structure, and we continue to make great strides in understanding the human genome and the importance of DNA to life and health.
So what did we actually learn from this discovery in 1953?
Scientists have since tweaked their findings a little bit, but overall, their model is still correct. DNA is a double-stranded helices, and the two strands are connected by hydrogen bonds. A bases are paired with T bases, and C bases are paired with G bases, which is consistent with and accounts for Chargaff's rules. Most DNA double helices are right-handed. That is, if you were to hold your right hand out with your thumb pointing up and your fingers curled around your thumb would represent the axis of the helices, and your fingers would represent the sugar phosphate backbone. Only one type of DNA, called Z-DNA, is left-handed. The DNA double helix is antiparallel, which means that the 5' end of one strand is paired with the 3' end of its complementary strand, and vice versa. Nucleotides are linked to each other by their phosphate groups, which bind the 3' end of one sugar to the 5' end of the next sugar. Not only are the DNA base pairs connected via hydrogen bonding, but the outer edges of the nitrogen-containing bases are exposed and available for potential hydrogen bonding as well. These hydrogen bonds provide easy access to the DNA for other molecules, including the proteins that play vital roles in the replication and expression of DNA. This is all we have today for this science lesson. Have a great day, class!